Hi, I'm Asmita Banerjee. We're back on this channel to talk about improving the quality of work in our cell culture labs. Why do we do research? Well, since prehistoric times, we humans have wanted to understand and explain things and phenomena that surround us. Oftentimes, our results are completely unexpected and contradict our hypotheses. In the lab, this often means that we do not know within which range our measured values are going to move. This makes it extremely important to cultivate the best possible practices and get a good training by an expert. Some researchers think they are safe, since they, anyway, use controls. But more often than not, these controls just tell us that the method and the device in the lab work. This can be highly misleading. Let me give you an example. A colleague was supposed to simply centrifuge blood samples and collect the supernatants. And this is what that colleague handed over to the technician. Measuring these samples together with the control serum will show that your control serum is in the correct range, but your measuring data and the interpretation will be completely wrong. What happened? First of all, the samples are not labeled. Second, some of the samples are hemolytic. This means the erythrocytes were lysed at some point and the hemoglobin got released. An unexperienced person will not recognize this, and even worse, will not know for what parameter it plays a role whether samples are hemolytic or not. For some parameters, hemolytic samples will give totally incorrect results. So, what went wrong here? Maybe the sample withdrawal was done incorrectly. Maybe it was done by an inexperienced or unsupervised person. Or the samples were centrifuged under wrong conditions and the erythrocytes were still in the supernatant. When you freeze a supernatant that still contains erythrocytes, they will burst during freezing, releasing hemoglobin. Or maybe the centrifugation was done correctly, but the supernatant was not removed in the correct way, and erythrocytes were carried over into the supernatant. See how easily data generation can go terribly wrong by a simple step such as collect the supernatant. This is why we want to contribute to improve the quality of work in our cell culture labs, and we want you to join us in this mission. I will see you in the next video.